Thank you guys for coming. I'm a little too excited. All right. Thank you for coming to this talk. This is my first time talking to a non-expert scientific audience. So thank you for being my guinea pigs, but you have an assignment, which is please feel free to interrupt with questions for me to explain anything that I reflexively am using as too much jargon or terminology. I'm going to do my best. But okay, so as you've already seen, the title of my talk is An Intuitive Guide to Weighing Evidence for and Against Universal Common Descent. This is a different talk, as you might notice, from the one that was posted on the schedule. And that's because I decided to spice it up a little bit. Okay, so the first thing we should talk about is this phylogenetic tree of life. How many of you guys have seen something like this? All life coming down in relatedness to a common ancestor through a process called descent with modification, which was pioneered by Darwin and others. And essentially in this tree, what you're seeing is that you have animals and plants and fungi and all kinds of bacteria that exist today are on what's called the leaves of the tree. They're related to each other through proposed or inferred ancestors at the nodes of the tree. And the length of these branches represents how different they are from their common ancestor. And so the most important thing, as I pointed out, is that the algorithm that builds this tree based on whatever properties you put into the computer program to compare amongst each other will always bring these elements down to a single root or infer that they came from one ancestor, okay? And so what I would like to talk about today is whether or not this hypothesis actually explains all the data, okay? So, but why should I talk about this at a Christian apologetics conference? For one, I want to just be clear that it's not required to reject universal common descent to be a committed believer in Christ Jesus. However, the evidence against universal common descent, or UCD as I may refer to it, is not widely known, even among scientists, people. This is so fascinating to me. It's like they're trying to bury the evidence. This imbalance leads to overconfidence in the UCD model and a misplaced belief that to reject UCD is an anti-intellectual approach based purely on the authority of a particular interpretation of the Bible. My goal today is simply to dispute that disposition with the facts and scientific argument. For those whose intuition tells them that when you look at a jellyfish and a giraffe and you say, no, there's just no way, that your intuition has a point. That's all I want to do. Come on in.
Trouble for bringing my flapjack.
Not at all. So before I get started, I'm going to describe what the evidence for common descent is, but to do that, I need to orient you to some background, and this is what's called the central dogma of molecular biology. Some of you may be familiar with it, and it's okay if you're not. We're going to learn a little bit about it right now. So you've probably heard the phrase DNA makes RNA makes protein. Potentially you've heard this, but if you haven't, that's good, because it's all wrong. It's nonsense. DNA is a molecule, and the molecule's not making anything. It sits there and it encodes something else for the whole system to read it out and to translate it to another language, the language of RNA. So what this is a molecule that is a polymer or has repeating units of A's, G's, C's, and T's. These are different types of components to the polymer, and they can be arranged in any order possible. Okay? And it's the sequence of this arrangement that encodes a particular protein through what's called translation. Once you have the messenger RNA, this is read out by, a protein uses DNA as a template to make a messenger RNA through complementarity. So all you need to know is that A is complementary to U, and T is complementary to A, and G to C. So you have GC base pairs and AT or AU base pairs. And now this serves as the starting point for the ribosome, which is this huge machine made of proteins and RNA. And they like to say it's very ancient, and that's because it's in all of life. That machine will read out this sequence, and every 3 bases here, these are called bases, the parts of the components of the polymer, is called a codon. And each codon corresponds to a particular amino acid, which is a component in another type of polymer called proteins. And these proteins fold up into unique shapes that give them their function and the things they do in the cell. Catalysis, like chemical reactions that help you break down your food and build up molecules you need, like for your bones, and basically everything, okay? Right, so here's the central dogma. So I'll be talking about sequences of portions of DNA, which we like to call genes, and these are just portions of DNA that have a particular function, or that will encode things with a particular function. Okay, so hopefully that gives us a little bit of a groundwork to go forward and talk about, well, what's all the fuss about? What is it that gives people the idea that universal common descent is a plausible hypothesis? Well, first, there's all kinds of similarity or homology across life, okay? So I'm showing you here a picture of like a physiological type of homology, right? So you have human, dog, bird, and whale, and they have arms. And within these arms, there are homologous sets of bones, okay, that do similar things, even though these arms are all pretty different on different animals. And so somebody called Darwin looked at that and was like, wow, how did that happen? That can't just be by accident that you came upon these features converged, let's say. That's very unlikely. The same thing can be said for when you dig down instead of the physiology, you look at those sequences of their genomes, and you see that you have homologous protein sequences or DNA sequences. And you can align them into a family, and you can see that in the positions of the polymer or of the protein, you have what's called you have conservation, okay? So you will put the same amino acid there almost all the time, and you see that a lot across the sequence, and people said, okay, if you can put, so I forgot to tell you, there are 20 amino acids. There's 4 bases for DNA, but there's 20 components to the protein polymer, and those can be in any sequence possible. So now you can imagine that if the space of the available sequences you can sample is really every position, you can put 20 there, you can put 20 there, you could put 20 possible there. That's 20 to the power of the number of positions in your sequence. So that is a lot. And so if you see this homology, it's like, well, if we had to choose these sequences, if life is choosing sequences at random, then it's more likely that they share ancestry and that they came from a common template somewhere in the past than that they arrived by accident at that similar sequence or similar physiology or whatever. So that's kind of what the basis of the argument is. And it's really a very, it's a solid reasoning, actually. The problem is the data don't all fit into this. So and that's what we're going to talk about today. So I'm going to show you, I like this. I found this video when I was studying, trying to learn about part of a theory of evolution for my work. And this guy who's going to talk, his name is Richard Lewontin, and he's like a really famous evolutionary biologist, and he's talking to historians of science about the debate in the past about how this particular part of the theory of evolution got popularized, let's say, is a neutral theory. And he's answering a question from the audience. It doesn't really matter what the question is. What's great is that he has this nice little picture of how he's going to answer the question, and I'll let you guys listen and then tell you what's important.
But John, look.
Can you hear?
Let's back to a basic model of organisms in the world. This is a kind of state space of organisms taxonomic space that was introduced by Keelan Hutchinson. And our observations are that organisms are distributed as clusters in taxonomic space. I mean, that's right. And there are clusters of clusters and clusters of clusters of clusters. And so they clearly have a relate. And the problematic for me for evolution is how do you explain the empty places? But that's another issue. Now, you're quite right that for Dubyansky, evolution meant, if you like, a movement of that cluster. Sometimes it meant a splitting of the cluster, two clusters. And sometimes it meant a movement of the cluster into space. But to describe it as a cluster, in some perspective, misses the point that the cluster is clustered someplace. And the problem of evolution, as often stated, is why did you start out here and get over here? So yes, it's true that it's the movement of the variation, but you still have to have a directionality to it. You
Okay.
still have to move the location of the cluster somehow.
Okay, so these clusters, each point represents an individual in a population, okay? And the reason, so taxonomic space, what he's saying is basically, so taxonomy is describing the relatedness of things based on their physical features. So there's all kinds of physical features, so you can put them in a multi-dimensional space, if you will, based on how those features vary amongst each other. And so now you have these clusters where the individuals in the group or the population, they're not identical twins, all of them, right? So there's like differences. But then if you compare them to the next closest group, they're like so far away compared to the differences among the members of each group, okay? And he is saying that, without really finishing the thought, if you notice, that this is the problem for evolution, is how do you get all that empty space between them, okay? Because evolution is supposed to be this real gradual process, and gradual processes don't leave empty spaces like that. It leaves, like, you know, members and populations in its wake. Or what you have to propose is that the gradual process happened, leaving no trace whatsoever. Like, everything in that empty space did live, and then it all went extinct. And when you do the math of population genetics, this actually becomes a really problematic proposal, because the number of individuals that have to die to create that empty space just could not have lived at that number could not have lived. It's just not what we see happening. So that's what I wanted to summarize for you, and that this is what there's some more probably complicated argument aspects to this argument, but I'm going to try and simplify it as much as possible to just say that it's the difference between the variance of anything. It could be the taxonomic, it could be the DNA sequence, whatever feature you're going to focus on. You look at the variance of that feature within a population, okay, and then you look at the distance of that correlated to that same feature to another population, and if that difference, the between group, is much, much larger than the within group, that is evidence, I'm arguing, for limited common descent, that they did not share a common ancestor. And so then what I will do in this talk is give you different features to pay attention to where that is like blaringly obvious. Go ahead, yeah.
Is this why the search for the missing link is such an important search?
Yes, absolutely. They know they need to fill those gaps, otherwise it looks kind of suspicious, yes. And where you look for those gaps is in the fossil record, right? If they just happen to not be in the fossil record, then what are we basing the proposal on, right? Okay. So just to clarify a little bit more, I'm going to be taking a page from geologists. So what do geologists do when they see a beautiful, very highly structured rock formation like this in Bryce Canyon? You have all these striations and things. When they want to date something, when they want to know something about the process that led to this kind of formation, what they have to do is look at processes ongoing in the present and to use those as a way to calibrate a sort of clock and apply that to the features they find in things that are old, where we no longer can see the processes ongoing, but infer what processes were ongoing to make those features in the past. And so you have weathering now. Let's say you have a river, and you can observe that over 5 years, this amount of sediment gets laid down from this type of a river motion, and this is the pattern that that sediment makes. And then, so now you have, okay, whenever we see this type of pattern frozen in an older rock feature, and it built up this many layers, then we know because it took 5 years to do this much, that's how old this rock is. So that's the kind of thing. The important thing is that if there is no process in the moment to observe, then that whole, your ability to do that kind of inference goes away. Okay, then you're just making stuff up, you see? Okay. Right. Okay, so I'll skip that for now. Right. So these are the types of features that I'm going to focus on, that where the differences are so big and so bad that it's like nobody even gets taught this in biology in college. That's how bad it is. Okay. I know this because I tell my students this, and they're like, "What?" So the genetic codes. The chromosome number differences, synteny, which is the fancy word for alignments of genes in equivalent places along the chromosomes, and then orphan genes. I'll tell you why the first 3 letters are capitalized when we get there. Okay, so the genetic code. Again, here's the central dogma of molecular biology. And what we'll be focusing on here is what I said before about these codons. There's a triplet code, right? And oh man, I could go on and on about how much design there is in the setup of this process, because it didn't have to be a triplet code. It could have been a quadruplet code. It could have been 2 letters to make an amino acid, but it's 3. And 3 gives you 64 possibilities for codons, and there's 20 amino acids. So you see now you have some opportunity for redundancy, which is what we have in the genetic code. And the organization of this redundancy is beautiful, but I'm not going to go into that. The point is, stay on track. Okay. So the point is that this is like a cipher, actually. It's a biological cipher. So in the cell, when you have this UUU, there is a little key that comes along and reads out UUU and says, okay, I have the right amino acid up here to go into the chain. And the thing is, I put "the" in quotes because it's not the only genetic code, it turns out. You can go to this website funded by our government to maintain in this database. There are at least 13 known genetic codes, okay? Going to be so much fun. There's 13 genetic codes, and okay, so somebody might argue, well, they're not that different, okay? There's like maybe 2 codons that mean something else now, and you have 64. So 2 out of 64, come on. That's basically the same. And that's essentially the argument that you see a lot, is like emphasizing the sameness, as if that's all you need to look at, because same means common ancestry, and then ignore the differences. Well, I'm going to do a little experiment with you guys to point out how devastating it can be to make tiny changes in that cipher, where you now reassign meaning to these codons. Here is a sentence where the meaning of 2 things has been reassigned. This is what the sentence used to be. Okay? All I changed was the 2 T's and the period. And I mean, when you first look at it, it's like gobbledygook, right? But that's the only thing that changed. So, all right, maybe the cell is smarter than we are at recognizing what the thing is supposed to say and can get by, right? Well, we're going to do a little experiment here. This is our evolution experiment. I want you all to take one of these. I'm going to try and pass them out. Take one of these and pass them. And don't look at it. until I tell you to. And what's going to happen in this experiment is you're going to turn it over all at the same time, and then you're just going to follow the instruction. That's it.
Are we being recorded?
I don't know.
Will this be on the final exam?
Yes. We need more. How many more? 2 more? Sorry. Okay. Oh my gosh. Okay. Here. Wait, they're supposed to be like kind of mixed up here, and they're not really that mixed up. Okay, there you go. You guys need more? Here you go. I think that pile's gonna come around there. Oh, sorry. Oh, this is exciting. Don't look at it yet! Cheaters. There's one in every population, even in biology. It's okay.
Which genetic code is that?
The sin one. Just kidding. All right. Okay, does everybody have one? All right, ready? Go. Turn over. Wait, if you get to get your Snickers as soon as you follow the instructions.
I gotta be honest, I didn't read the last part.
Okay, we can stop now because the food has run out. Okay, what I'm gonna do, how many of you guys, okay, yes, go back. I'm sorry, there's no more food for you. How many of you who have a Snickers had the one that was spelled totally weird? 1, 2, 3.
Were they not all spelled totally weird?
They were not. Right? You are a mutant, okay? Your genetic code changed. Right, exactly. Okay, come on in if you want. Oh, I don't know. Anyway, so the thing is, this is the point, is that the minority of people who were able to get a Snickers had the genetic code switch. The majority of people who got it had the wild type or the normal genetic code. So even though it's not impossible for it to work, you have to compete with the people who didn't have that switch. And then you have to explain why the people that didn't have the switch just got extinct because they didn't have the switch. That just doesn't fit, right? That's a disadvantage. In limited resources, which is always the case outside of the lab, you are not going to do something that's so incredibly costly to your entire system and get away with it. So that's what I wanted to say about the genetic code. There have been some, so people have published, articles and done research to look into this. And I won't go into the details of this particular one, but what I think is very, incredibly interesting about this particular paper is if you map the instances of these weird genetic codes on existing trees that you can build using other features, it doesn't map in a way that makes sense at all. Like, you would build a different tree if you started out with the genetic code as the feature you were building the tree based on. So you get tree mismatch. And since we all would, so anyway, then you have to come up with elaborate, apologetics really for these trees to try and make the model fit. And so that's what's happening in this paper, and it's really, I don't know. I go back and forth between laughing to myself and being like, why don't you see it? So, Okay, so that's one. Okay, so in theory, oh yes, okay, so then somebody might ask, because it is kind of like, well, they are really similar, and how did that happen, by accident, if it happened, to happen independently over and over again, right? And this, of course, is operating on the assumption of materialism, which you don't have to take, but they do. So this is the question they have, right? And why should the codes be so similar at all? Because if you can imagine, you can completely scramble all the codes of every single independent lineage, and then it would be like, well, yeah, they have nothing to do with each other. Maybe someone would say that, right? But there's several lines of evidence that suggest that the organization itself of the genetic codes is a feature that promotes robustness and survival of the organism. So it's not just like it could have been anything. There's something really important about the way it's organized. So of course you'd want to preserve that across lineages. And then because the lineages are not identical, something changes within the organization of the organism. And so this is one of the things. Okay, so chromosome number, maybe I should stop and say, does that kind of make somewhat of sense, hopefully, to some of you? Okay, yeah.
Just as a non-biologist, let me ask a question to see if I'm understanding.
Sure, please.
Are you saying that if we look at the genetic code between species, if we look at the similarities, there's a lot of similarities?
Yes.
If we look at the differences, there's more differences than there are similarities?
No, the reverse. There are more similarities than differences. But what I'm claiming is whatever process that you might propose to even make those tweaks would be so devastating to the cell that it's irrational to propose they happened. And then the explanation for the similarities is that there's common constraints on the system that those similarities are answering to support the biology, the similar biology. Or just like, they're alive. And I'll probably explain more about that later. Okay, so chromosome number is another one of these big problems, in my opinion. And every time somebody asks me, like, oh, or somebody claims two things that seem really similar, like rats and mice, for example, come from a common ancestor, the first thing I do is I go to Wikipedia. And there's this cool list in Wikipedia of all the chromosome number counts for a bunch of different organisms that you can look up. So here I'm showing you, just to sort of establish the point of why this is important, one example where you can actually sort of cross the species barrier where there's a chromosome number difference between horse and donkey, and probably a lot of you know you can get a mule from that, but maybe not a lot of you know that mules are pretty much infertile. So, yeah, 64 is the number of chromosomes that, and this is like, so it's going to be 32 pairs really. So that's what they look like, and then you have 62 for donkey. Yeah, so it's not impossible, but again, with the experiment that we just did, just because something is possible doesn't mean it's like the great explanation, like it's the most plausible explanation for the data. Okay, so, and then I'm just going to show you, mice and rats have 40 and 42 chromosomes, similar to donkeys and horses. So I would expect this to be a problem as well for what would the common ancestor, if he or she, if they existed of rats and mice, how many chromosomes did they have? And then what was the process that led to either a loss of a pair or a gain of a pair? So I think what's hard about this is that this is not something that people, I think, do experiments on to actually test, right? And so a lot of whether you feel that this is a problem or not is based on like how much intuition you have about like genetics and all of that. So,
Question.
Yeah.
People have been breeding mules for 3,000 years.
Breeding mules
Yeah.
together.
Well, horses and donkeys. They've been
Oh.
making mules for farming
Right.
for thousands of years.
Mhm.
Many large numbers of farmers have been doing. Wouldn't there have been, am I but they're not? Is that what you're going towards?
Yeah, I think the fact that like human intervention is continuously required to make mules is a good argument for the fact that there was never any common ancestor that had 63 chromosomes.
Millions of farmers for thousands of years making horses with donkeys to make mules.
Right. Yeah, and this is not a sustainable process. You have to keep on inputting, and that's because of the infertility. If it had a chance of becoming, like, you would think after this amount of time, I agree, that the population of mules, there would have been some kind of change that would have allowed them to just like mate with each other and then just become a self-sustaining population, but that hasn't happened. And I think that is because there are mechanisms to create barriers between groups that had an independent emergence. Yeah, go ahead.
So I noticed in the mule had, was it 63 versus the 62 and the 64, which have been even, which had an even split.
Yeah.
So is that probably the extra chromosome almost not because it's not paired? Is that what causes the infertility at that point?
Yes. It makes meiosis. Have you guys heard of mitosis and meiosis? Awesome. So, to make the gametes or the sexual reproductive cells that only have one copy in them, so you reduce the copy by half in each generation, so that sexual reproduction brings it back to the normal number. So one of the horse chromosomes doesn't have an equivalent pair in the donkey set, and so that's going to be. Yeah, exactly.
Real quick question, real simple. Chromosomes versus DNA. We always get this picture of DNA as being this beautiful long double helix. Is it in fact broken into segments?
Yes, the segments. Well, no, the segments are the chromosomes. So the whole chromosome is one long double helix continuing for like millions and millions.
The whole DNA molecule resides on each chromosome. Got it.
Yeah. The chromosome, what it is it's like, I think they said if you stretched the human genome, it would go to like the moon or something crazy like that. Even though like the polymer itself is microscopic, well, not even microscopic, it's like way smaller than that. And so how does it get condensed into the cell is because there are proteins that essentially wind the DNA around it like a bobbin, like thread around a bobbin kind of thing, which is amazing. And then in order to read out information, it has to get unwound selectively at different parts, and all of that is highly regulated. It's just so crazy. This gives me chills. This is why I do what I do. I just love it. Yes?
Is there a similar issue with the mutation of chromosomes, such as in like a Down syndrome?
Oh my gosh, yes. Okay, thank you. I really should have... I forgot that example exactly. This is in the human example. Here is another part of the evidence for why chromosome number matters. The genomes are very sensitive. Okay, you can, for the most part, when you have it's called like a segregation, I think, problem. So in mitosis, as you remember, you pull the sister chromatids apart. Everything has to go equally into each side, but sometimes 2 go over here and 0 goes over here into the other cell. So you would get like trisomy, so you have a copy, an extra copy of a particular chromosome. Now, for the majority of the chromosomes if that happens, there's just spontaneous abortion of the growing fetus in there, because it's like that devastating, and there's only like a couple or a few that you can actually get a live birth from that. And then there are, of course, birth defects and developmental challenges associated with people who have that. And then you ask the question, are those people fertile? And the answer is not so much, and I don't even know if Down syndrome people, I feel like they would have to like mate with each other in order to have a chance, because you have the same problem as the mule situation in terms of having in this case, extra genetic material that has no pair. So basically, meiosis would be very, very confused and messed up, and And yeah, and that is a huge evolutionary disadvantage and a problem that there's not like a mechanism to just deal with. So you can do this over and over again throughout life and just be like, yeah, now let's have a new species with this number of chromosomes. So it's just, there's no mechanism for that. Yeah.
I assume this is a different mechanism, but the fusion and splitting of chromosomes, such as the most common theory right now where humans came from is that chimpanzee chromosome 2A, 2B fused create humans.
Mm-hmm.
And my problem with that is you need to have the chimpanzees close to each other. You need to create a male and female human, and then they have to procreate. I find that highly unlikely.
Right?
So
Yes.
that's a different mechanism.
I wasn't even going to go into that because I was like, oh, nobody's going to like, you know, but yeah. I feel like I don't want to like say this on the record because it's not coming from my scientific expertise. It's more just like a cross between, you know, my personal theology hypothesis, but that is a problem. Okay, just for all of you to kind of be on the same page. So chimpanzees and bonobos and gorillas have 48 chromosomes, and how many do we have? 46, right? So we have 23 times 2, right? And they have 24 times 2. So that is like based on the argument I'm making, a huge evidence that there's no common ancestry there. But some people found that the signatures, I wish there was a chalkboard in here. So there's signatures meaning like special patterns in the DNA sequence at the tips of the DNA, and there's special patterns at the center that are important for the function of being able to pull them apart during mitosis or even to finish replicating the copy all the way to the end of the chromosome. And they have names that I won't like belabor. In any case, it looks like those special sequences are in the middle instead of at the end. Well, not instead of, in addition to at the end, there's some of that in the middle of our chromosome 2, which makes it look like another chromosome fused and to like two chromosomes fused, and then there's some scrambling so it doesn't look perfectly like the way the original signature would look, but it retains some of the same functional features, and so people are like, there you have it. That shows that in the past, you know, we had a common ancestor that had 24 times 2, and then fusion happened in, I don't even know how to finish that sentence, right, actually. How many individuals did that fusion happen in? And, I forgot your name, Kevin? Like Kevin was bringing up, and I brought up earlier, there's a problem when you don't have a matched set when you have sexual reproduction. So you would have to have the fusion happen in a male and a female in their gametes, and those gametes have to meet together, and then their kid would be 48 and they would both be 46. Like, that's just, okay, so here's what I think happened. Sorry. Don't throw me out of here, but I do think that, I think we started out, I think we were created with 48 chromosomes. Sorry, what's the one that they have? 48, right? Yeah, 48. I think we were created with 48 chromosomes. And I think at some point in our history, it was decided that there needed to be a really strong barrier between our species and other primates. And I think that happened because of something else that happened in our history, which is interbreeding between us and other non-humans that created hybrids that were very problematic for God's plan. And I think God said, you guys are gonna try this crap again, and I can't have this. Like, I got the rainbow, I can't do the same thing again. So I'm just going to nip it in the bud. And if you think that it's implausible that we would try that, there are scientists, I think they're in Italy, who were actually proposing to try and make a hybrid between human and chimp in the lab.
Maybe in China
I'm sure that there's, I mean, it's crossed all of our minds. I'm not going to just be like, oh, I'm holier than them. I want to know, honestly, if it would work, but something stops me, which is that I think it's anathema. Okay, go ahead.
So if that was successful, would they have an odd number if that weird species who would have that odd number?
It would, yes. And
We don't have a species that has that middle odd number.
there isn't one on the planet.
It's a similar analogy to the mule.
Exactly. And so even if we did do it and it worked, that would not be its own lineage that would go on, and most likely, just like there's not a freestanding mule lineage. And that's why I think that happened, is because that's just,
If we could have made a freestanding mule lineage, somebody would have done it.
sure. It's a lot less work probably than breeding. Exactly. Yeah. I think there was another question. Go ahead.
Yeah, this is kind of going back to the when you're talking about the Down syndrome and that being losing a chromosome some one way or the other. How does that happen? Because it does happen.
In the Down syndrome case, you have an extra one. Yeah.
An extra one. So you have an extra one. What causes that?
It's like any mutation, these are chemical reactions happening at the speed of light, essentially, like basically literally in the cell. No chemical reaction is, perfectly complete or does the same exact thing every time. And so I feel like, the answer is that it's just happenstance, That's not a very satisfying answer, probably, but.
We're not exactly sure exactly why it happened.
Yeah, I mean, it's related to the age of, I think. I don't know if they're debunking that now with more newer data, but I think it's related to the maternal age of conception and things.
So if the mother is older, there's a greater chance.
Yeah. Mm-hmm.
Thank you very much. I'm trying to come up with counter arguments.
Oh, please.
I'm thinking back to my 9th grade biology class, and we were taught about punctuated equilibrium.
Yeah.
So what's the guy's name? The
Is it? Yeah, I know.
Is that cool?
Stephen Jiggle. Mm-hmm.
Anyway, so the clusters on the board. So here we have a cluster over here. How do you explain the gap?
Yeah.
And so what do you think of this? So here's how it explains. Populations are usually stable.
Yeah.
And then every once in a while, some event happens or something. And triggers a kind of leap. And then we get out of it a new cluster. Right? And so that's why we don't have that kind of slow continuity.
Mm-hmm.
Punctuated equilibrium. What do you think about that?
Yeah, I mean, I kind of feel like the punctuated equilibrium, the reason why it fell out of favor is because people were upset that it sounded like creationism. Right, they never proposed a mechanism for how you get this leap, right? You have to explain, yeah, it's just, I don't know, I'm not doing a very good job of debating this point. But yeah, I mean, later in that same video that I put up, there's another historian who points out that one of the scientists in the past was proposing that you had to have a genetic revolution in order to get that drive to a new part of feature space or whatever, taxonomic space, but that same scientist never proposed what would cause such a revolution. So that's sort of an adaptationist view, what you're saying. So there's like these different schools. There's the school where like most changes or mutations are neutral, or they do nothing bad or good, and then a few of them are advantageous, and some of them are deleterious, but the vast majority are neutral. So basically, populations are just drifting around in this neutral space where you can look a little different, but it's not any worse than this guy. Look a little different, but you have this little change in your metabolism, but it's not gonna, it's not really helping you. It's just, for kicks. And then a boulder falls on half the population, and these guys survive only because they didn't get crushed by the boulder. And now those changes that were in them move on to the next generation. So that's what we have today, or what explains the diversity is just what's called drift, or random chance that we see it, not because those changes are actually beneficial, all of them, right? And then you have the adaptationist view, which is that when you see a difference between, any two populations, it's because there was some kind of natural selection that, allowed them to adapt to a new thing and all, so probably I would say the punctuated equilibrium argument would rest on an adaptationist view that, there was some kind of catastrophe and boom, now you're very minority part of the population is adapted to that new environment and everybody else dies and that there you left a gap. You have some okay, this is what I don't like about that idea though, is that we don't see it happening right now. Okay, that's the thing that I said you can just make stuff up if you don't have the river doing the thing right now. Like if you don't see that kind of catastrophic, change happening in the present, then you're just making up that it happened in the past and there's no way to verify that. I don't think that's a very strong historical scientific method personally. So it's not impossible, but it rests on about as much ongoing observational proof as creation. We don't see creation happening right now, do we? Unless we're doing it ourselves in the lab. It fell out of favor relatively quickly. And yeah.
I got an A on the test in high school. I don't know if I need to go back and take it again.
No, I mean, I think it's good it's being taught and that's the thing that's so, I'm gonna wax philosophical here about pedagogy, is how you teach stuff, right? That you teach that there's a debate, there has been a debate throughout the history of evolutionary theory. And that helps you learn what are the strong points and the weak points of all these different arguments, except there's just a total exclusion selectively of some parts of the debate that just become the elephant in the room and it's just obnoxious. So why not just add that to the debate, and how much stronger would the teaching of the whole discipline be? In my opinion, it would be very strong, but I think, you might run the risk of people, kids recognizing that there are too many glaring holes in all sides of the debate on one side for certain proposals, I should say. And one of them would be universal common descent, I think. So yeah, evolution is great. It's just how many starting points were there? That's the question, I think, is the most important. All right, so let's go. I have this decision tree. This is a little caveat. Okay, so what I do when I go to this Wikipedia page, I say, okay, do they have the same chromosome number? Oh, okay, if they do, then that's not going to be evidence against universal common descent, potentially. But if they don't, then I have to ask, do the numbers differ in a way that's not a factor of a whole number? So meaning, would it be this organism has 10 chromosomes and this organism has 20 chromosomes? I'd be then I next would ask, okay, are they plants? Because what happens in plants is you might get a mistake, sort of like with the Down syndrome case we were talking about, where you don't pull the two sides apart equally, but you actually end up having a fusion where you end up with a whole duplicated, an extra set of chromosomes. And now that's a new lineage, but they aren't infertile in plants. Plants are very malleable genetically for some reason, and they can handle polyploidy is what it's called. And so a lot of bread, corn and things like that, have multiple copies of genomes inside their cells compared to their ancestors. So that, I would say, is not evidence against the common descent of those two things if they have that. But if it's some random, one has 10 and one has 17, then I'm gonna be no. Okay, and so I would say common ancestry is unlikely in that case. So this is kind of how I do this decision tree. And I love this website. There are organisms, it's listed in order of the number of chromosomes. And the amount of genetic material that some of the tiniest things that we would say are just super simple, protists or things floating on the scum of the ocean or whatever, that we were they don't do anything. They have so much more genetic material than us by 5 orders of magnitude or something. And the idea of more information in terms of just sequence equals more complexity really does not follow. I just think that's an interesting fact. Okay, so Oh. Well, we're not going to do this one. All right, so synteny or genome alignment. Are we like ready to go? Yep. Shoot. Okay, yes, let's skip this one. Or not, I'm not going to skip it. I'm just going to go really fast. And you can come up and ask me questions later. I'll be around. Okay. This is essentially when you have, okay. Let me give you my analogy. Okay, so we have a deck of cards, and you know what's inside a deck of cards. And we're going to have a machine that templates the production of another deck of cards based on the what's inside of the first one, okay? And it does it in order. And then you have a second deck of cards that has the same exact order. But if I gave you, I had 2 decks of cards here, but it's okay because we don't have time. And I was going to show you that they're kind of in the same order, but then like not really. There's some switching happening, and then you're like, what happened to your machine? I thought that it was supposed to just go in order, and every time I have an ace of spades over here, I'm supposed to have an ace of spades over here. So in this analogy, let's say like the cards represent genes, and so they're supposed to be in a particular order on the chromosome, and then you have another group of organisms where they have kind of all the same genes, but they're just not in the same order. And this is not talked about as a problem, but it is, because DNA replication doesn't switch things around. Mutations do, or like other mechanisms, and there's a bunch of mechanisms, but what I wanted to say is that a lot of them are dangerous for the cell. And so here's one example with rats and mice again, okay? What you're looking at, all of these columns, each one has 2 sides to it, and there's colors that represent the genetic material that is homologous to, these are rat chromosomes, and this column is mouse, and this column is human. Okay. And then each color represents the chromosome on mouse or human, where that genetic material of the rat is homologous to. Okay, so the point here, all you need to know is that if there was no switching around of chunks of DNA, all these would have like one color, all right? That's what it would be. But you see that it's not. And you would expect that maybe for the human comparison, because we're further apart from rats than mice are. But even as close as you might think rats and mice are, look at all of this. How do you explain that? I mean, the closest one here, this is a sex chromosome, so that's interesting. This is a really close one. But all of this is like just totally scrambled, okay? So that's another problem. And the problem really is that if you don't see that scrambling within the population, then it's like right now, if you don't see a process ongoing that creates that that type of variance where you would be able to explain how you could survive, then this represents a giant gap, and gaps are bad. Okay. So the last one is orphan genes, and this is the spiciest one. I should have put it first. I'm sorry, guys, but basically, I've been talking about homologous sequences this whole time, and that's like the strongest evidence, right? All genes come from older versions of genes, okay? That's sort of the paradigm. But then what do you do when you have sequences of DNA that code for protein, that do important things in the cell, that don't look like anything else in the tree of life anywhere? Okay, these are called orphan genes because they have no family. You can't group them with others like them in other organisms like you can with like almost a lot of proteins. But the thing is that it turns out these are so pervasive, they've been under our nose the whole time, and we have been throwing them out as some sort of like error of sequencing because they didn't know how to annotate them. They didn't know what they were. And so they were like, this is a mistake. They're not a mistake. They do things like in the mouse one, sorry, I just explained that. So the point is that there are orphan genes between rats and mice. There are genes that are unique to mice. And some of these even are involved in the production of sperm. Why am I saying that? Because it's not like these things are just like, oh, they're kind of useful, but you could do without them. You can't do without the production of sperm. So whatever process is being proposed as an ongoing source of new genetic material within species, it has to explain how the species survived without that gene until now. And then it also has to convince me that this is a process. We need to, again, look within populations. And if you see within a subset of the population orphan genes that the rest doesn't have, so like there's some mice and they have this new sequence and we don't know what it does yet, but whoa, where did it come from? Well, and they're able to interbreed, of course, with the rest of mice, then I would be convinced that this is a process ongoing, as opposed to evidence that they were just different from the beginning with a different lineage, yeah. It's related to that, but it's a different thing, yeah. So anyway, those are the 4 lines of evidence, and I have this cute, very like, these are provisional conclusions, by the way. This is my caveat too. More data has to come in, as I described, and what we need to do is just sequence everything, and then everyone will know. And that's the end of my talk here. This is dramatic for effect. This is not fair, but anyway. Yeah, thanks for all your questions, and I'll be around if you have more, so thanks.